Okay, good afternoon. Today we are going to continue with the course. So just to remind you where we are still in the cellular level of organization. We started with studying the cell. We studied the cell membrane last time, and today I'm going to go into the inside of the cell, the cytoplasm. And the cytoplasm has the cytosol and has organelles inside it. So the cytosol is the intracellular fluid, and it constitutes about 55% of the size of the cell. The remaining 45% is actually the size of the organelles, because the cytoplasm is cytosol and organelles. So 55% cytosol, 45% organelles, and the cytosol is the a jelly-like substance consisting of fluid. 90%, up to 90% of it is fluid, which has dissolved components inside it, dissolved particles, either organic or inorganic particles dissolved in it, and it is the site where many chemical reactions are taking place, either to produce ATP or in order to synthesize the building blocks. By the word building blocks, for example, the protein synthesis, which is synthesized from small blocks. So a protein molecule, for example, is mainly synthesized from amino acid molecules. These amino acid molecules are present in the cytoplasm, like here the large organic molecules are amino acids. In addition to that, there are some cytoplasmic inclusions. These are particles which are not dissolved. They are in organic molecules. And these particles, they are not present in all cells, but depend on the function of the cell. Like a fat cell, which stores fatty acids, lipids, in the form of a big droplet inside the cytoplasm. As you can see it here, it occupies most of the size of the cytoplasm. Or in some others, like in the cells of the liver, they store glycogen. Glycogen is a large molecule of carbohydrate and is used for storage of carbohydrate, which provides energy for the body. Some of the cells, like the melanocytes of the skin, as we will see next week, they have melanin pigment inside them to protect the cells from the effects of ultraviolet radiation. So you find melanin pigment inside the cell. These are called inclusion bodies. So again, I repeat, we are studying the cell. We studied the cell membrane last time. Today, we're studying the cytoplasm. The cytoplasm has the cytosol, which is mainly fluid, in addition to these particles dissolved in it, plus the organelles. So now I'm going to concentrate on the organelles. So these are either membranous structures or non-membranous, surrounded by a membrane or not surrounded by a membrane. Like the filaments, that's microfilaments, which constitutes the part of the cytoskeleton of the cell, they are not surrounded by the membrane. When an organelle is surrounded by a membrane, then this membrane has exactly the same structure of the cell membrane. Remember that it has phospholipid, phosphorus, and fatty acids here, and then on the other side we have, so there is a bilayer of phospholipids, and then there are the protein molecules here. Some carbohydrates are present. So it's the same structure. When I say membranous, it's the same structure of the cell membrane. And some of the organelles have one membrane surrounding them with a double layer of phospholipids. Like, for example, the vesicles. We talked about them last time, or the lysosomes. We're going to talk about them this time. Some of the organelles, they have double membrane. So they have double of the double lipid bilayer. And these, for example, the mitochondria or the nucleus as well. So the nucleus, in some textbooks, it is just regarded as one of the, just another organelle, or because it's very important for the function of the cell, as it controls the function of the cytoplasm and contains the hereditary material. So they just give it some sort of focus and study it as separate from the other organelles, but in fact, it's one of the organelles. Most of the cells have nuclei. Actually, all the cells have nuclei. But some of the cells, they have nuclei during part of their life. and then they lose their nucleus, like what happens here, for example, in the red blood cell. The red blood cell is manufactured, is produced in the bone marrow, and then when it becomes mature, it will be pushed to the circulation. And as it is pushed to the circulation, it's going to lose its nucleus and become anucleate. That's why, because there is no nucleus, it cannot repair itself, and it cannot replicate. So that's why it has a lifespan of 120 days, and then it's going to be destroyed because it cannot repair itself. And the reason for that, it has no nucleus. We will mention that again when you study the cardiovascular system. The reason is that to make it have a very big cytoplasm, to increase the oxygen carrying capacity of the cell. That's why sometimes they call them not red blood cells, they call them red blood corpuscles, because they are not actually cells at that time. They don't have a nucleus. On the other hand, some other cells, they have multiple nuclei, like as you can see here. And this is we're going to study it in the last lecture of this block. This is a muscle cell, skeletal muscle cell, and you can see that there are multiple nuclei here in this type of cell. So either no nucleus, or there is usually one nucleus, or there are multiple nuclei. And it is one of the organelles that has a double membrane, and it is perforated by pores, as you can see it here. Nuclear pores through which substances pass between the nucleus and the cytoplasm, including like small molecules or large molecules, ribosomes, messenger RNA, as we will see in a moment. Inside the nucleus is the genetic material. The entire genetic material is called the genome, and the genetic material is arranged in chromosomes. I'm not going to go into details of the chromosomes and the genes. This is out of the scope of this course, but just to let you know that there are 46 chromosomes, or 23 pairs of chromosomes in somatic cells, because we have two types of cells in the body. We have somatic cells, and we have the gametes, or the sex cells, like the ova, the oocyte, and the sperm. So these cells, the gametes, they have half the number of chromosomes. When they meet together, they will have the complete number of chromosomes, the diploid, 46, 23 pairs of chromosomes. And each chromosome, as you can see, is formed of chromatids. It's a very long coiled tube of chromatin. So it has chromatids, which is a long coiled tube of chromatin. Each chromatin has coiled around some proteins, which are called histones, and it's formed of deoxyribonucleic acid mainly, so DNA mainly. And as I said, it controls hereditary material, transmit hereditary material from one generation to another, and it controls the activity of the cytoplasm, the production of proteins in the cytoplasm. And the proteins, they give the identity of the cell. They control the function of the cell. They are part of the enzymes that are produced by the cell. They are part of the hormones by which the cell is affecting other cells. So it's very important. Inside the nucleus, there is a dark area which has no membrane called the nucleolus, and it is the site of production of ribosomes. You will see that these ribosomes are produced in the nucleus, specifically in the nucleolus of the nucleus. Then they are transported through the pores of the nuclear envelope. We say nuclear envelope because it's not a single membrane, it's a double membrane. You can say nuclear membrane, but it's a nuclear envelope indicates that it's thicker than that. So it passes through the nuclear membrane or nuclear envelope and reaches the cytoplasm. In the cytoplasm, it is the site of protein synthesis. So the site of protein synthesis, which is present in the cytoplasm, is produced in the nucleolus of the nucleus. So these ribosomes, in fact, they are formed of RNA, ribonucleic acid, and protein. They are packages of RNA and protein. They are assembled in the nucleolus. They go as the site of protein synthesis. They are either present free in the cytoplasm, where they are going to manufacture, where they're going to produce the proteins that are used inside the cell, or these ribosomes are attached to a network of flattened sacs, which are called the endoplasmic reticulum. So that network will look as if, because it has multiple ribosomes, it looks rough. and so it's called rough endoplasmic reticulum. And these ribosomes are responsible for the production of proteins that are either going to be used in the cell membrane to replace pinched parts of cell membrane, or to be exported outside the cell, like hormones or enzymes being exported outside the cell. So the ribosomes are produced in the nucleus, pass through the nuclear envelope, reach the cytoplasm, where they are present in two locations, and they have the same function, but the product is different. The function is protein synthesis. So you can see here that what happens, in fact, is that during protein synthesis, this double helix of DNA will separate, and a template in one location of what we call a messenger RNA will be formed. And then this messenger RNA will pass from the nucleus through the pores, reaches the cytoplasm. In the cytoplasm, the messenger RNA, which you can see it here as a strip messenger RNA, this will act as a template to produce or to manufacture amino acids. So for example, this messenger RNA carries codes. Let's say here 101 and then 000 and 111. These are the codes for amino acids. And then the amino acids are present in the cytosol. They are dissolved. And so they should be brought from the cytosol, a specific amino acid, because we have 20 amino acids. So we have to bring the amino acid 010, which fits in here. And this is brought by what we call transfer RNA. So there's a transfer RNA that will carry the amino acids from the cytoplasm, take it to the messenger RNA where it fits. The messenger RNA is attached to the ribosome, okay, whether it was on the endoplasmic reticulum or it was free in the cytoplasm. So now we have amino acid 1, and then amino acid 2, then amino acid 3, and then a chain is formed of amino acids. A chain of amino acids means protein. So that's how the protein is formed. It is coded by the messenger RNA, and the messenger RNA is coded and controlled by a process which is called transcription, which is in the nucleus, depending on the chromosomes. Here, this process of coding is called translation. So from transcription to translation, a special sequence of amino acids is formed, a special protein is formed, and this was under the control of the nucleus. So that's how the nucleus controls the function of the cell. I'm not going to go into details. You probably have this in some other course. So the endoplasmic reticulum, as I mentioned, it's a network of sacs, and this network of sacs, as you can see, is connected to the outer layer, outer membrane of the nucleus. Said there are two membranes, so the outer membrane connected to it, it's a network of flattened sacs, and these flattened sacs, if ribosomes are attached to them, then it's called rough endoplasmic reticulum. If it has no ribosomes, then it is smooth endoplasmic reticulum. So the rough endoplasmic reticulum, the one with contained ribosomes, means that it is the site for protein synthesis. That's what its function. It's the site of synthesis of protein and the beginning of what we call processing of protein. The protein is not a simple chain like I drew it right now, like multiple beads in a chain. It's not like that. It is, to start with, it's like that, but then these beads are going to be folded in a 3-dimension. And so this 3-dimensional representation, it depends also the properties of the protein. In addition to that, some of the proteins have things added to them, like, for example, glucose to form glycoproteins, or lipids to form lipoproteins, or, for example, in the hemoglobin, iron is added to it. So multiple things are added to it. It's not just a simple chain of amino acids. This is called processing. And the processing of protein starts in the endoplasmic reticulum, in the rough endoplasmic reticulum, but the main site for processing of protein is the Golgi apparatus, as we will see in a moment. The smooth endoplasmic reticulum has no ribosomes. It's smooth, has no ribosomes, Therefore, no protein synthesis. So what is it for? It is for the synthesis of fatty acids. So when fatty acids and steroids are produced here in the smooth endoplasmic reticulum, cells like the cells which constitute the adrenal cortex that secrete sex hormones, or cells in the testis that secrete, which are steroid hormones, they have a lot of these smooth endoplasmic reticulum. In addition to that, the smooth endoplasmic reticulum in other cells which do not synthesize these hormones, it detoxifies harmful substances, for example, alcohol, or some drugs like phenobarbital. These drugs, they are toxic, so they are detoxified in the sedatives. They are sedatives, and they are detoxified in the smooth endoplasmic reticulum. And when a patient starts taking these drugs more and more, the amount of smooth endoplasmic reticulum increases, so that more of the drug is detoxified, and the patient will not feel the effect of the drug until the lethal dose is reached and causing death. The other function in some other cells, especially in the muscle cell, and we are going to discuss this in details later on, is that the muscle cells, skeletal muscle cells, they have a very complex smooth endoplasmic reticulum which stores calcium ions. These calcium ions are important for muscle contraction. At one time in muscle contraction, you need a gush of calcium ions, and then this calcium ion should return back, should disappear later on. So they will be stored in the smooth endoplasmic reticulum, and in the muscle, the smooth endoplasmic reticulum has a special name. It's called sarcoplasmic reticulum. The Golgi complex, again, it's a group of flattened sacs. So they are bundled together like a stack of pita bread, and they are flattened, curved membranes. They are sacs. It is the place where most of the processing of protein will take place. So the proteins that are produced in the ribosomes, they will be transported by transport vesicles, and here they are going to be processed. Again, I mean, by processing is acquiring three-dimensional shape, adding other molecules to them like lipids or sugar or ions added to them. And so they are processed there, and then they are packaged, sorted out, packaged. This one should go to the plasma membrane. This one should be exported. This one is bad, so let's take it to a lysosome, and so on. Okay, because during the manufacture of proteins, this is a continuous process, and like any other factory, faults can take place. So this faulty protein has to be destroyed as well. And most of the destruction will take place in the lysosomes, although the protein itself, in fact, takes place in other vesicles which are called proteasomes. I'm going to talk about them later on. So let's deal with lysosomes to start with. They are membranous vesicles, single membrane, and they are formed in the Golgi complex. They are filled with digestive enzymes, the enzymes that are going to attack, engulf bacteria. Remember last time when we talked about endocytosis and phagocytosis? So they digest the large molecules and microbes, and also they recycle own worn out organelles. These organelles, some of them are worn out, so they can be digested by the cell itself, or they digest entire cell after death. What happens after death is that these lysosomes will explode and spill the digestive enzyme into the cytoplasm and will digest itself. Sometimes they digest substances outside the cells, like this is what happens in the sperm, for example. The sperm, as we will study it in the block 3, has a head. Most of the head is occupied by the nucleus, and then just on top of the nucleus, capping the nucleus, if this is the head of the sperm, this is the flagella or the tail, and then you have a nucleus here, and then on top you have a cap. This is a very big lysosome called acrosome, and so this lysosome will open, secrete, expel the digestive enzymes, and these digestive enzymes will digest, at one point, will digest the the cells and the wall that surrounds the ovum, so that when there's a pore is formed, it's going to push the nucleus in, and the two nuclei will unite together in the process of fertilization. These are peroxisomes. They are like lysosomes. They are vesicles. They contain enzymes, digestive enzymes, but they are smaller than the lysosomes. They contain enzymes, two types of enzymes, either oxidases, which break down fatty acids and produce hydrogen peroxide, H2O2. That's why they are called peroxisomes, because they result in the formation of hydrogen peroxide. Okay, this is a cytotoxic product and is used to detoxify many chemicals, toxic chemicals, including alcohol. So alcohol is detoxified in these peroxisomes, and also it is detoxified in smooth endoplasmic reticulum as well. And so you can see here, for example, this is an electron microscopic view showing you one of the liver cells in a rat, normal rat, and then another rat, which is exposed, has been injected with a toxic chemical. You can see here that this rat will develop a lot of these peroxisomes in order to fight and destroy these toxic substances. It also contains enzyme called catalase that decomposes the excess of hydrogen peroxide, because hydrogen peroxide by itself is lethal to the cell. So we don't want excess of this hydrogen peroxide, and so it is the same peroxisome that produces the hydrogen peroxide as a byproduct. It decomposes it. Proteasomes, and these proteasomes destroy the proteins, the cytosolic proteins, either unneeded extra protein, damaged protein, or faulty protein during the process of protein synthesis. They have found in research that these excess of these damaged or unneeded protein or faulty protein is present in the cytoplasm of neurons of people with Parkinson's disease and Alzheimer, and it's probably this is one reason that one cause of this disease. We don't know exactly. This is because these cells, they have defective proteasomes, so they cannot attack, they cannot remove the faulty protein, and this will be deposited in their cytoplasm. The other organelle is the mitochondria. So the mitochondria has a double membrane. This is one of the organelles that has double membrane, mitochondria and a nucleus. It is sausage-shaped, as you can see it here, and it has a central cavity, which is called the matrix, and the inner membrane has multiple folds. You see here, these are multiple folds of the inner membrane. The inner membrane here, the yellowish one here. So that's the inner membrane, and the matrix inside it. And the idea is, with the folding, it increases the surface area, which is available for chemical reactions. Once you see folds, then you think of increasing the surface area of something. You'll see that in, for example, in the microvilli. I'll show it to you in a moment. This is to increase the surface area for chemical reactions. These chemical reactions are needed to produce the ATP, the energy that is required for the cell. That's why they are considered the powerhouse of the cell, because they are the site of main site for production, generation of ATP, and the enzymes that are needed are present in the matrix, inside the matrix, and they work on these folds, which are called cristae. So it plays an important role in apoptosis, which is programmed cell death. Some of the cells are programmed to die at a certain time, and at this time, it is the mitochondrial enzymes that are present in the mitochondria will be released and cause the cell death, or start the cell death, and then the lysosomes will take over. And it contains some DNA. This DNA is important for the synthesis of mitochondrial proteins that are required for the production of the ATP. So this DNA, in fact, it comes from the mother, doesn't come from the father, because during fertilization, it's only the nucleus that is donated or that passes from the sperm to the ovum. The ovum has a nucleus and the cytoplasm containing the mitochondria, and these mitochondria are maternal mitochondria, not paternal. So we inherit more genes from the mother than from the father. But it's only a small amount of genes that is present in the mitochondria. Like we have about 30,000 genes, but only probably 37 of them are present in the mitochondria. But yet we inherit more genetic material from the maternal side than the paternal side, because we do not inherit mitochondria, or we do not receive mitochondria from our fathers. Now, inside the cytoplasm, there are non-membranous organelles, and they form what we call the cytoskeleton, the skeleton of the cytoplasm. They are a network of proteins like filaments, as you can see them here. They are like filaments, threads. So they are with three types: microfilaments, as the name indicates. They are very thin and small, or they are intermediate, or they are in the form of microtubules. Proteins, which are called tubulins, they arrange together and form a very narrow tube. So that's why they are called microtubules. And they serve as a scaffold to support the cell shape, organize some chemical reactions, and they are important in the motility, either motility of the entire cell or part of the cell, like the cilia or the flagella of the cell, as we will see, or movements that take place in the cell. For example, movements of chromosomes that take place during cell division. The microfilaments, as the name indicates, are the thinnest, and they are present at the periphery. They are important for scaffolding, giving the shape of the cell. They pass through microvilli, which are multiple finger-like processes from the apical surface of the cell. What do you think that these finger-like processes function? Multiple finger-like processes from the apical surface of the cell. What is the function of the microvilli? So they increase the surface area. So that's why you find them, for example, in the cells of the intestine, the small or large intestine. All of them, they have microvilli to increase the surface area of the cell membrane for absorption. They are non-motile, they do not move, but they just increase the surface area. The microfilaments are also present in muscle cells. We're going to talk about them when we deal with the muscles. They are formed of actin and myosin, and these are types of proteins that are important for the contractility of the muscle. Intermediate filaments are formed of several proteins. They are larger, and they are important for anchoring organelles or attaching one cell to another. And then we have the microtubules, which are larger cylindrical structures composed of tubulin, as I mentioned, and they are very important for the movement of chromosomes during cell division and movement of cilia and flagella. They are produced in another organelle in the cell, which is called the centrosome. It's located very close to the nucleus. This is the site where these microtubules are manufactured. And as I said, they are very important in cell division. So in cells that do not divide, like the nerve cells, they don't have centrosomes, because you don't need that organelle. There's no cell division taking place. This is the centrosome. Here we have other processes arising from the cell membrane called cilia and flagella. Differentiate between cilia and flagella and the microvilli. These are the microvilli. I have just mentioned about microvilli. They are from the apical surface of the cell, finger-like processes. This is an electron microscopic view of an apical part of the cell. You can see here there are mitochondria. These are some lysosomes, probably peroxisomes, and these are the microvilli, finger-like processes. They can only be seen, they are not the villi that you can see them under the light microscope. They are microvilli, so you can only see them under electron microscope. Under light microscope, this is the villi of the small intestine. Under light microscope, you will find that there is some sort of fuzzy appearance on the apical surface of the cell. It's not that sharp, it's a little bit fuzzy. So that's why they call it the brush border. They do not appear as finger-like projections, but they appear as fuzzy edge of the cell, and it's called the brush border of the cell. So when you hear brush border, then think of microvilli, and as I said, they are non-motile, they are multiple, and they increase the surface area. What maintains these finger-like projection is the threads of cytoskeleton, which is present inside the cell. So that's why they are not compressed, because they have cytoskeleton inside them, as you can see here. These are the microfilaments. They maintain their shape. As I said, they maintain the shape of a cell. Now, this should be differentiated from cilia and flagella. So the cilia, they are motile projections, okay? And they are composed of bundles of microtubules. And the cilia, usually they are short and multiple. And they move. They have a sweeping action. So that's why, for example, they are present in the lining of the respiratory passages. And here they sweep the particles, the carbon particles, the dust particles, they sweep them away from the lung, and then and we cuff them after that. And then there is a single one which is very long, and it's called flagellum or flagella. They are long and single, motile. And the best example is in the flagellum of the sperm, and it moves the entire cell. These, they do not move the cell, they move by themselves, but they don't move the cell. So that's the difference between cilia, flagella, and microvilli. They are modifications of the apical modification of the cell membrane. Yep.
Just a question. You said it was always example with sperm, tail of sperm. Is there another example in the actual human body?
I don't recall right now. Yeah, I think it's only the sperm.
Okay.
Yes, it's only the sperm, the flagellum of the sperm. I don't recall any other example. Yeah. Okay, so I'll skip this slide. Do you have any questions? Thank you for your patience, and we'll continue next time.